Welcome to Bedtime Science. Did you know that the internal clock ticking inside a woman's brain runs about 6 minutes faster than the one inside a man's? And that tiny difference quietly reshapes when they feel alert, when they dream, and when rest finally arrives. Tonight we explore the quiet biological rhythms that make men and women surprisingly alike and subtly different. In the next moments, we will gently look at how internal clocks differ by just minutes, why the moon-like rhythm of hormones reshapes the architecture of the night, how muscles respond almost identically regardless of sex, how puberty sets different developmental tempos in boys and girls, and how light itself is processed differently behind male and female eyes. Before we begin, if you find comfort here tonight, stay tuned for more updates. And if you're still awake, you can leave a soft comment about what topic we should visit next. Now let's begin. Deep inside your brain, behind the bridge of your nose, two small bundles of nerve cells sit just above the place where your optic nerves cross. Together, they form a structure called the suprachiasmatic nucleus. Most people have never heard of it, but it's been quietly governing your life since the day you were born. This structure is small, about the size of a grain of rice. It holds roughly 20,000 neurons, a tiny fraction of the brain's 86 billion. And yet these 20,000 cells act as the body's central timekeeper. They set the pace of your day. They decide when hormones rise and when they fall. They shape when your body temperature climbs and when it drops. They choose when alertness sharpens and when it fades. Each cell in this cluster generates its own rhythmic electrical signal, a pulse that repeats roughly once every 24 hours. When these cells fire together, they produce a coordinated wave of activity that ripples outward through the brain into the rest of the body. That wave is your circadian rhythm. The word circadian comes from two Latin roots, circa meaning about, and diem meaning day, about a day, not exactly a day. And that small imprecision turns out to be one of the most telling differences between male and female biology. In 2011, a research team led by Gene Duffy at Harvard published findings in the Proceedings of the National Academy of Sciences. They measured the intrinsic circadian period, the natural length of one full cycle of the internal clock, in a group of men and women living under carefully controlled laboratory conditions. No windows, no clocks, no time cues of any kind. What they found was a small but consistent gap. Women, on average, had an intrinsic circadian period of about 24.09 hours. Men averaged about 24.19 hours, a difference of roughly 6 minutes. 6 minutes. You could lose 6 minutes looking for your keys, but inside the circadian system, 6 minutes changes everything. About 35% of the women in the study had an intrinsic period shorter than 24 hours. Their clocks ran slightly fast, always pulling them toward an earlier schedule. Among men, only about 14% had a period that short. Most male clocks ran a little slower, drifting gently toward a later schedule each day. Here's the thing. That 6-minute difference in the clock's natural cycle doesn't simply produce a 6-minute difference in behavior. It amplifies. Through the complex feedback loops of melatonin secretion and light sensitivity, that small gap in period length translates into a roughly 30-minute earlier onset of melatonin in women compared to men. A fourfold magnification. From 6 minutes of clockwork to half an hour of hormonal timing. This is why many women tend to feel drowsy a little earlier in the evening. It isn't preference, It isn't habit. It's the physics of a biological oscillator running at a slightly different speed. And this small structure, this grain of rice-sized cluster, even differs in its physical shape between the sexes. In females, the suprachiasmatic nucleus tends to be more elongated. In males, it tends to be more spherical. Same organ, same function, but built in slightly different proportions, like two versions of the same quiet instrument, tuned just a few notes apart. One of the hormones that may explain why the female clock runs faster is estradiol, a form of estrogen. In animal studies, estradiol has been shown to shorten the circadian period. When estradiol levels are high, the clock ticks a little more quickly. When they fall, the clock slows. This means the female circadian system isn't fixed. It shifts across the menstrual cycle, across pregnancy, across menopause. The clock's speed is partly governed by the hormonal tides moving through the body at any given time. The male clock, by contrast, runs at a more consistent pace. Testosterone follows a simpler 24-hour cycle, peaking during the night and declining through the day. It doesn't reshape the clock's period the way estradiol does. It modulates, it adjusts, but it doesn't rewrite the tempo. So here, at the very beginning, we find a pattern that will repeat throughout tonight's conversation. Men and women share the same fundamental machinery, the same suprachiasmatic nucleus, the same neurotransmitters, the same melatonin pathways. But the hormonal environment surrounding that machinery creates subtle, persistent differences in how it all behaves. Two clocks, same design, different calibration. Both ticking quietly right now, inside you, as you listen to these words. The suprachiasmatic nucleus doesn't work alone. It needs information from the outside world to stay synchronized. And the most powerful piece of information it receives is light. Light enters the eye and strikes a specialized set of cells in the retina. Not the rods and cones you learned about in school, but a separate population called intrinsically photosensitive retinal ganglion cells. These cells contain a blue light-sensitive pigment called melanopsin. When light hits them, they send a signal directly to the suprachiasmatic nucleus along a dedicated neural pathway called the retinohypothalamic tract. This signal tells the clock what time it is in the outside world. But here's what's interesting. The clock doesn't respond to that signal the same way in everyone. Research has shown that females tend to exhibit greater melatonin suppression and a stronger alerting response when exposed to moderate or bright light at night. If a man and a woman are both exposed to the same amount of light, say a brightly lit room at 11 in the evening, the woman's melatonin production will typically drop by a larger amount. Her clock is more sensitive to the signal. The molecular basis for this appears to involve estrogen. When estradiol is present, it enhances the expression of certain proteins in the suprachiasmatic nucleus, specifically C-phos and phosphorylated CREB. These proteins are part of the signaling cascade that resets the clock in response to light. Think of it this way. Light arrives at the clock like a knock on the door. In both men and women, the door opens. But in women, especially when estradiol is circulating at higher levels, the door opens a little wider. The signal gets in more easily. The clock shifts more readily. Males, on the other hand, have higher expression of androgen receptors in the core region of the suprachiasmatic nucleus, the area where retinal input arrives first. Testosterone tends to blunt the photic phase-shifting response. It doesn't block it, it dampens it. The knock still lands, The door just doesn't swing as far open. This has practical consequences. When researchers measure dim light melatonin onset, a standard marker for circadian phase, men consistently show a later onset than women. And when exposed to the same intensity of nighttime light, men show a smaller degree of melatonin suppression. The two clocks are reading the same sky, but they're interpreting it differently. And these differences aren't static. In women, the sensitivity of the circadian system to light appears to fluctuate with the menstrual cycle. When estradiol is high, around ovulation, the system responds more strongly to nighttime light. When estradiol is low, the response diminishes. The male system, driven by a steadier hormonal backdrop, shows a more uniform response across days and weeks. So the light that enters your eyes right now, the glow of a screen, the warmth of a lamp, the faint blue of a winter evening, is being processed by a system tuned not just to brightness, but to the hormonal conditions inside your own body. The retina gathers the light. The melanopsin absorbs it. The signal races along dedicated fibers to the clock. And the clock, shaped by estrogen or testosterone, elongated or spherical, fast or slow, decides what to do with it. Two sets of eyes, looking at the same world, building slightly different pictures of when the day should end. And somewhere in that difference, a few minutes of phase, a few degrees of sensitivity, the quiet gap between a man's evening and a woman's evening is written, not in choices, not in habits, but in the molecular grammar of light and time. Consider this. There's a rhythm in female biology that has no equivalent in male physiology. It cycles not every 24 hours, but roughly every 28 days. And it's so ancient it predates our species by hundreds of millions of years. The menstrual cycle is what scientists call an infradian rhythm, a biological clock whose period is longer than one day. The word comes from the Latin infra, meaning below, because its frequency falls below the once-per-day threshold of the circadian system. This roughly 28-day cycle is driven by a cascade of hormones: estrogen, progesterone, follicle-stimulating hormone, luteinizing hormone, each rising and falling in a pattern that governs far more than reproduction. It reshapes the structure of the night itself. During the mid-luteal phase, the second half of the cycle after ovulation, when both progesterone and estradiol are elevated, something shifts in how rest unfolds. Research consistently shows that during this phase, women experience more wake after sleep onset events, more brief arousals, lower overall efficiency. The night becomes more fragmented, not dramatically, not in a way most women would describe as insomnia, but measurably. The transitions between stages grow a little less smooth. The periods of deep recovery shorten by small increments. The continuity of the night frays at its edges. Progesterone plays a central role in this reshaping. It increases what's called REM latency, the time it takes to enter rapid eye movement stages after first falling into rest, and it reduces the total proportion of REM across the night. You get less of it. It takes longer to arrive. But here's where the mechanism gets nuanced. Progesterone itself isn't simply disruptive. Its metabolites, the breakdown products created as the body processes progesterone, act as positive modulators at GABA receptors. GABA is the brain's primary inhibitory neurotransmitter. When these progesterone metabolites bind to GABA receptors, they enhance the receptor's activity, producing a sedative-like effect. So progesterone promotes non-REM stages through its calming metabolites, while simultaneously making the transitions into REM more difficult. It pushes the architecture of the night in two directions at once, more of one kind, less of another. During the mid-luteal phase, elevated progesterone also increases the density of fast spindles in the second stage of non-REM. These spindles, brief bursts of rhythmic brain activity between 12 and 16 Hz, are associated with memory consolidation. They appear to play a role in how the brain files away the experiences of the day. More spindles, potentially better consolidation, but less REM where a different kind of memory processing occurs. The night becomes a different machine during this phase, still functional, still restorative, but reorganized like a library where the books have been moved to different shelves. And then comes the late luteal phase. Progesterone drops, estradiol falls, subjective quality deteriorates even further. Women in this phase report more awakenings, more restlessness, a greater sense that the night simply wasn't enough. This isn't imagined, it's hormonal. The architecture of rest is being rebuilt and torn down on a roughly monthly cycle, following a blueprint that has nothing to do with stress, or caffeine, or screen time. It's the body's own 28-day construction project, raising and lowering the scaffolding of the night according to a plan written deep in the endocrine system. Men, by contrast, experience none of this cyclical restructuring. Testosterone follows its simpler 24-hour rhythm, rising during the night, peaking in the early morning hours, declining through the day. It tracks existing REM cycles. It doesn't substantially reshape the architecture from one night to the next. The male night is more consistent, not better, not more restful, just more uniform across days and weeks. And so here's one of the most profound rhythmic differences between the sexes. Not in how much rest is needed, that's roughly equivalent. Not in the fundamental cycle of non-REM and REM, that's shared. But in the month-to-month variability of how that cycle is built. A woman's night is a moving target. A man's night is a more fixed point. Both functional, both ancient, both doing the same essential work, but doing it to different rhythms, like two tides shaped by different moons. The differences we've walked through so far, in clock speed, in light sensitivity, in hormonal reshaping of the night, might suggest that men and women are biologically quite different creatures. But if we pull back and look at the larger patterns, a different picture shows itself. Women tend toward an earlier chronotype. They're more likely to be what researchers call morning types, feeling alert earlier in the day, preferring earlier bedtimes, waking more easily in the early hours. This tendency increases with age, and it's more pronounced in women than in men across most of the lifespan. But the autonomic nervous system, that vast unconscious network governing heart rate, digestion, breathing, and the seesaw between arousal and rest, operates in both sexes on the same fundamental principles. The sympathetic branch activates during moments of alertness and effort. The parasympathetic branch restores and quiets. This wiring is identical in men and women. Reproductive hormones modulate how this system behaves. They adjust the set points. They shift the thresholds, but they don't redesign the system itself. Both estrogen and progesterone affect thermoregulation, the body's ability to control its own temperature. And temperature is one of the primary signals the circadian system uses to promote the transition into rest. When core body temperature drops in the evening, It triggers a cascade of events that prepare the brain for its nightly work. In women, particularly during the luteal phase and around menopause, the hormonal influence on thermoregulation can increase nocturnal awakenings, hot flashes, night sweats, brief spikes in skin temperature that pull the brain momentarily toward wakefulness. These same hormones affect respiratory control. Progesterone is a respiratory stimulant. It increases ventilatory drive. During the luteal phase, women breathe slightly differently during the night, not dramatically, but enough that respiratory patterns shift in measurable ways. Men's relatively stable hormonal environment produces more consistent patterns from night to night. The thermoregulatory system runs on a steadier track. Respiratory patterns hold closer to their baseline from one week to the next. But here's what matters most. Despite all these differences in clock speed, in light response, in hormonal cycling, in thermoregulation, in respiratory drive, the total need for rest and the fundamental structure of the non-REM to REM cycle remain remarkably similar between men and women. The same number of hours, the same basic oscillation between deep recovery and vivid neural activity, the same homeostatic pressure building during wakefulness and dissipating during the night. The differences are real, measurable, physiologically significant. But they're variations on a theme, not separate themes entirely. Like two rivers flowing through different terrain, carrying the same water, obeying the same gravity, arriving at the same sea. And now we come to a part of the story that may surprise you. Because when we leave the brain and move to the muscles, to the body's largest organ system, the differences between men and women become even harder to find. Dr. Lauren Colenso-Semple, a researcher who studies sex differences in exercise physiology, has said something that challenges a lot of popular assumptions. Men and women, she noted, show very similar protein metabolism responses to resistance exercise, and very similar growth responses. Think about that. When a man lifts a heavy weight and when a woman lifts a heavy weight, the molecular events that follow, the breakdown and rebuilding of muscle protein, the signaling cascades that trigger growth, the adaptation of muscle fibers to load, are nearly identical. The narrative that women need special sex-specific training programs is, according to the data, largely unsupported. As Colenso-Semple put it, the data says men and women respond to exercise very similarly. So where do the visible differences come from? If the muscle tissue itself responds the same way, why do men typically carry more mass? The answer lies not in the muscle, but in the hormonal history of the person carrying it. During male puberty, testosterone surges to levels roughly 10 to 20 times higher than in females. This surge drives an increase in lean body mass, bone density, and muscle fiber cross-sectional area that persists into adulthood. The muscle tissue itself isn't fundamentally different. It was simply exposed to a different hormonal environment during a critical developmental window. The baseline difference in mass is a product of history, not of ongoing biology. And there's another layer to this. Standard hormonal assays, the blood tests commonly used in clinical settings, are not capable of detecting the lower levels of circulating testosterone found in women. The tests simply lack the sensitivity. This measurement gap has obscured our understanding of female hormonal baselines for decades. We've been trying to read a signal with a tool that can't detect it. When exogenous androgens, synthetic testosterone or similar compounds, are introduced into a female body, something revealing happens. Female musculature develops to levels that rival non-steroid-using males. The capacity was always there. The machinery was always the same. It was waiting for the hormonal signal to arrive. What this tells us is something profound about the nature of biological sex differences. Many of the visible differences we see between men and women aren't differences in hardware. They're differences in the chemical signals that activate that hardware. The same engine, different fuel mixtures. A muscle fiber building itself back after effort doesn't know whether it belongs to a man or a woman. It knows only the signals it receives, the tension it endured, the nutrients available, the hormones circulating in the blood. It responds, it adapts, it grows. The same quiet process in every human body, repairing itself in the dark. The differences between men and women don't appear all at once. They emerge slowly over years, during one of the most turbulent periods of biological development, puberty. But even here, in this phase of dramatic change, the rhythms of development differ between the sexes in ways that are often overlooked. Dr. Catherine Page Harden, a behavioral geneticist, has studied these differences extensively. And one of her key findings concerns a distinction that most people never think about: the difference between pubertal timing and pubertal tempo. Pubertal timing is when puberty begins. Pubertal tempo is how quickly it unfolds. For girls, Harden's research suggests that early pubertal timing, starting puberty at a younger age, is the best predictor of a wide range of outcomes. Mental health difficulties, physical health challenges, earlier menopause, even in some studies, a shorter lifespan. Starting early changes everything. For boys, the picture is different. Emotional and developmental outcomes appear more closely tied to pubertal tempo, not when the changes begin, but how long they take to complete. A boy who moves through puberty rapidly faces a different set of challenges than one who develops gradually, regardless of when the process started. Two different rhythms, timing for one sex, tempo for the other. This is a deep structural difference in how development unfolds. It suggests that the male and female bodies aren't simply running the same program at different speeds. They're running different programs with different critical variables, different sensitive periods, different points of vulnerability. And yet, both programs accomplish the same essential task. They take a child's body and rebuild it into an adult one. They activate the hormonal systems that will govern circadian timing, metabolic function, and reproductive capacity for the decades that follow. The pubertal surge of testosterone in boys creates the muscle mass differences we talked about a few moments ago. The pubertal maturation of the hypothalamic-pituitary-ovarian axis in girls sets up the monthly hormonal rhythm that will reshape the night for the next 30 or 40 years. These aren't small adjustments. They're foundational changes, and they're governed not by a single event but by a rhythm, a developmental rhythm that has its own pace and its own rules, different in boys and in girls. The interaction of genes and life experience during this period shapes long-term health in ways we're only beginning to understand. Early pubertal timing in girls doesn't just change adolescence. It sets a trajectory. It alters the speed at which the biological clock will run for the rest of her life. And the tempo of puberty in boys doesn't just affect the teenage years. It establishes patterns of hormonal regulation, emotional reactivity, and physical development that persist into middle age and beyond. These adolescent rhythms are the origin story of the adult body. Every circadian difference, every hormonal pattern, every metabolic tendency we've discussed tonight traces back to this period, to these years of quiet, relentless biological construction. Puberty isn't a single event, it's a rhythm. And like all the rhythms we've examined tonight, it runs differently in two versions of the same human body. One keyed to timing, the other keyed to tempo. Both building, both becoming, both arriving at adulthood through different doors. And somewhere in the memory of your own body, that rhythm still echoes, the developmental tempo that shaped you, the timing that set your course, still reverberating in the way your clock ticks tonight. Now let's look at what happens when the seasons change. Because the body doesn't only keep time with the day and the month, it keeps time with the year. Seasonal rhythms, circannual rhythms as researchers call them, influence hormone production in both men and women. Testosterone levels in men show subtle seasonal variation, tending to be slightly higher in autumn and early winter. Estradiol levels in women also fluctuate with the seasons, though the patterns are complex, intertwined with the menstrual cycle in ways that make them difficult to isolate. These yearly rhythms are still being mapped. Researchers know they exist. They can measure them. But the full picture, how seasonal variation in light exposure interacts with reproductive hormones to modulate the circadian system differently in each sex, remains an area of active investigation. What's clear is that the body contains nested cycles, rhythms within rhythms within rhythms. The heart beats roughly once per second. The circadian clock turns roughly once per day. Hormonal cycles run on schedules of weeks. Developmental rhythms unfold over years, and seasonal rhythms repeat across the calendar of a lifetime. Each of these rhythms influences the others. The circadian clock modulates heart rate. Hormonal cycles reshape circadian timing. Puberty sets the hormonal patterns that will drive both circadian and monthly rhythms for decades. Seasonal variation adds yet another layer of modulation on top of it all. Men and women share every one of these timescales. Every one. The differences lie in the details, in the specific hormones that drive each rhythm, in the amplitude of each oscillation, in the sensitivity of each system to external signals. But the architecture of timekeeping is the same. And there comes a point in every life when the most dramatic hormonal shift of all occurs. For women, it's menopause. For men, it's the slower, more gradual decline sometimes called andropause. These are very different processes. Menopause is an abrupt transition. Over the course of a few years, estradiol and progesterone production falls steeply. The monthly cycle ceases. The hormonal patterns that have been reshaping the night for decades go quiet. Research shows that during menopause, falling estradiol and progesterone contribute to hot flashes, night sweats, and awakenings. The suprachiasmatic nucleus, which contains estrogen, progesterone, and androgen receptors, loses a significant portion of its hormonal input. Circadian rhythms grow less stable. Complaints of fragmented rest increase. Hormone therapy can partially normalize circadian parameters, suggesting that restoring some hormonal input to the clock and to thermoregulatory centers restabilizes the system. The clock can still respond to its old signals. It just needs them to be present. In aging men, the process is slower. Testosterone declines gradually over years and decades. There's no single moment when the hormonal environment overhauls itself. Instead, it erodes slowly, steadily, like a coastline retreating from the sea. This gradual decline in testosterone and changes in androgen signaling within the suprachiasmatic nucleus are linked to reduced circadian amplitude, increased daytime drowsiness, and more fragmented nights. The clock keeps running, but its signal weakens. The peaks grow lower, the valleys shallower, the rhythm flattens. And here's where the story comes full circle. By late older age, both sexes show more similar patterns: lighter rest, more fragmentation, reduced REM, reduced slow-wave activity, earlier melatonin onset, earlier habitual bedtimes and wake times. The 6-minute gap between the male and female clock, it narrows. The difference in light sensitivity, it diminishes. The monthly reshaping of the night that made female rest a moving target, it ceases entirely. The clocks converge. After decades of running at different speeds, tuned by different hormones, shaped by different cycles, the male and female circadian systems begin to resemble each other more closely than they have since childhood. A 2024 review on sex differences in circadian rhythms notes that sex is a critical variable earlier in life. But in older adults, the common aging-related changes, the shared decline in amplitude, the shared fragmentation, the shared advance in timing, become the dominant features. Menopause and andropause become modifiers rather than primary drivers. The body ages toward a kind of convergence, a place where the differences matter less and the shared biology matters more. Two clocks, once set at different speeds, now slowing together. Like two instruments that played in different keys for most of the concert, and in the final measures, find themselves playing the same note. Let me gather the remaining threads of this story and continue through the details that fill in the picture we've been building. The suprachiasmatic nucleus receives input not only from the eyes, but from the rest of the body. Temperature signals, hormonal signals, signals from the gut. It integrates all of this information and sends timing signals outward to peripheral clocks, smaller timekeeping mechanisms embedded in nearly every organ and tissue. Your liver has a clock, your kidneys have a clock, your skin, your lungs, your fat tissue, all of them contain molecular oscillators that cycle roughly once per day, synchronized by signals from the master clock in the brain. In women, the monthly hormonal cycle creates a shifting relationship between the master clock and these peripheral clocks. When progesterone rises in the luteal phase, some peripheral clocks may drift slightly out of alignment with the central clock. The body's timekeeping grows a little less coordinated, a little more complex. This isn't dysfunction. It's a feature of a system designed to accommodate a reproductive cycle that requires different organs to operate on different schedules at different times of the month. In men, the simpler hormonal cycle keeps the master clock and peripheral clocks in a more stable relationship. The coordination between central and peripheral timing stays closer to baseline from week to week. Both systems are elegant. Both accomplish the essential task of keeping the body's organs working in temporal harmony. But one does it with a steady hand. The other does it while dancing. Let's talk about body temperature for a moment, because temperature is one of the most important signals the circadian system uses. Core body temperature follows a daily rhythm. It rises through the morning, peaks in the late afternoon and drops through the evening. That evening drop is one of the triggers that initiates the transition into rest. In women, this temperature rhythm is modulated by the menstrual cycle. During the luteal phase, baseline temperature rises by about 0.3 to 0.5 degrees Celsius. This is a well-known phenomenon. It's the basis for some natural family planning methods. But that elevated baseline means the evening temperature drop starts from a higher point. The system has to fall further to reach the threshold that triggers melatonin release and the onset of drowsiness. In some women, this makes the transition into rest slightly more effortful during the second half of the cycle. In men, the temperature rhythm is more consistent. It follows the same daily arc, day after day, without the monthly elevation and return. During menopause, the thermoregulatory system becomes unstable in a different way entirely. Hot flashes, sudden surges of heat that typically last 1 to 5 minutes, disrupt the temperature rhythm unpredictably. They can occur during the day, but when they happen at night, they pull the brain toward wakefulness, fragmenting the continuity of rest. The underlying mechanism involves the narrowing of the thermoneutral zone, the range of core body temperatures within which the body doesn't need to actively heat or cool itself. Declining estradiol appears to narrow this zone, so that smaller fluctuations in temperature trigger sweating, flushing, and increased blood flow to the skin. The body becomes more reactive to its own heat. A temperature change that would have gone unnoticed at 35 now triggers a full vasomotor response at 55. And yet, men in later life also experience changes in thermoregulation. The circadian amplitude of the temperature rhythm decreases with age in both sexes. The evening drop becomes shallower. The morning rise gets less pronounced. The signal that tells the body when to transition between wakefulness and rest grows quieter. Different paths, same destination. The temperature rhythm slowly flattening, like a wave losing energy as it approaches a distant shore. Let me tell you about melatonin itself. This molecule that has been woven through our story from the very beginning. Melatonin is produced by the pineal gland, a tiny structure deep in the center of the brain. The pineal gland sits outside the blood-brain barrier, which gives it direct access to the bloodstream. When the suprachiasmatic nucleus signals that darkness has arrived, the pineal gland begins synthesizing melatonin from serotonin through a two-step enzymatic process. The melatonin rises, it enters the blood, it reaches receptors throughout the body, and it carries a simple message: night is here. In women, as we discussed, this melatonin onset occurs about 30 minutes earlier on average than in men. But the total amount of melatonin produced over the course of the night is roughly similar between the sexes during the reproductive years. What differs is the timing and the sensitivity to suppression by light. After menopause, melatonin production in women often decreases. The loss of estradiol appears to reduce the pineal gland's synthetic capacity, though the mechanisms are still being studied. This decline in melatonin is one of the factors that contributes to the increased difficulty with rest that many women report after menopause. In aging men, melatonin production also declines, but more gradually, in parallel with the slow decrease in testosterone. By the 7th and 8th decades of life, melatonin levels in both men and women are substantially lower than they were at 20 or 30. The signal of night grows quieter with age. In both bodies, at roughly the same pace, once the acute hormonal transitions of menopause are past. Melatonin does more than signal darkness, by the way. It's also a powerful antioxidant. It crosses cell membranes easily and scavenges free radicals, the reactive oxygen species that can damage DNA and cellular structures. Some researchers believe that the age-related decline in melatonin contributes to the accumulation of oxidative damage that characterizes aging itself. A molecule that tells the body when to rest also protects the body while it rests, and its decline with age may be part of why the body becomes more vulnerable as the years pass. The same molecule, in men and women, doing the same work, fading at the same pace. Now let's think about cortisol, the other great circadian hormone. Cortisol is produced by the adrenal glands, which sit on top of the kidneys. Its rhythm is almost a mirror image of melatonin's. Cortisol stays low during the early night, begins rising in the second half, and peaks in the early morning, about 30 to 45 minutes after waking. This morning peak is called the cortisol awakening response. In women, the cortisol rhythm is modulated by reproductive hormones. During the luteal phase, baseline cortisol levels tend to be slightly elevated. During pregnancy, cortisol rises steeply. By the third trimester, levels can be 2 to 3 times higher than in non-pregnant women. Men show a more stable cortisol rhythm across days and weeks. The amplitude is similar. The timing is similar. But the modulation by reproductive hormones is less pronounced. Chronic stress can flatten the cortisol rhythm in both sexes, reducing the morning peak and elevating nighttime levels. When this happens, the circadian system loses one of its most important timing signals. The body has difficulty distinguishing between day and night. Rest becomes harder to achieve and less restorative. And here's something worth knowing. The interaction between cortisol and the immune system follows a circadian pattern. Cortisol suppresses inflammatory responses during the day, and as cortisol falls in the evening, inflammatory markers rise. This is why fevers tend to spike at night, why joint pain often worsens in the evening, why wounds may ache more after dark. This pattern is the same in men and women. The circadian modulation of immunity operates on the same clock, with the same rhythm, producing the same nightly rise in inflammatory signaling. The differences are in degree, not in kind. Women's immune systems tend to mount stronger inflammatory responses overall, a trait that provides better defense against infection, but increases the risk of autoimmune conditions. Men's immune systems tend to be less reactive, which reduces autoimmune risk but makes them more vulnerable to certain infections. But the rhythm is the same. The clock that governs when the immune system ramps up and when it stands down ticks at the same pace in both bodies. Let's turn our attention to the brain itself now, to what happens inside the skull when the body transitions from wakefulness into rest. The electroencephalogram, the EEG, shows us the electrical rhythms of the brain. During the transition from wakefulness to the first stage of non-REM, a distinctive pattern emerges. The fast, low-amplitude beta waves of waking consciousness give way to slower, higher-amplitude alpha waves, then to the even slower theta waves of early non-REM. This transition looks remarkably similar in men and women. The same frequencies, the same progression, the same gradual deceleration of neural activity. Where differences appear is in the deeper stages. Women tend to have more slow-wave activity, the deep, high-amplitude delta waves that characterize the most restorative stage of non-REM. This holds true during the reproductive years, and it persists to some degree even after menopause, though it declines with age in both sexes. More slow-wave activity doesn't necessarily mean better rest. It means a different distribution of the night's resources, more time in deep restoration, potentially less time in other stages. Men tend to spend slightly more time in the lighter stages of non-REM. Their architecture distributes rest differently across the stages. And when it comes to REM, the stage of vivid dreaming, rapid eye movements, and muscle paralysis, the differences are small. Both sexes cycle through REM periods that grow longer as the night progresses. Both experience the characteristic sawtooth waves on the EEG. Both show the same patterns of brainstem activation and forebrain activity. The dreams of men and women are built on the same neural scaffolding. Spindles, those brief bursts of rhythmic activity in the 12 to 16 Hz range, appear more frequently in women's EEGs. We mentioned earlier that progesterone increases fast spindle density during the luteal phase. But even outside of luteal phase effects, women tend to generate more spindles overall. Spindles are thought to serve as a kind of protective mechanism. Brief moments when the thalamus gates incoming sensory information, preventing external stimuli from reaching the cortex and disrupting rest. More spindles might mean a more effective gating mechanism, a stronger defense against nighttime disruption. And yet, paradoxically, women report more subjective complaints about the quality of their rest than men do. More difficulty falling into it, more awakenings, more dissatisfaction with the night. This is one of the great puzzles in this field. By many objective measures, women's rest architecture is equal to or even slightly better organized than men's. But subjectively, women report worse experiences. The disconnect may have hormonal roots. The monthly cycling of progesterone and estradiol creates variability that may register as instability in subjective experience. Or it may have cultural roots, different expectations, different pressures, different relationships with the night. Or it may be something we don't yet understand, a gap between the brain's electrical activity and the mind's experience of it. A place where measurement and perception don't quite agree, like two maps of the same territory, drawn in different languages. The brain is an organ that burns a striking amount of energy, about 20 watts at rest, roughly the power of a dim light bulb. It weighs about 2% of the body's total mass, but consumes about 20% of its energy. During the night, the brain doesn't simply shut down. It shifts into maintenance mode. One of the most important maintenance functions is carried out by the glymphatic system, a network of channels surrounding blood vessels in the brain that flushes out metabolic waste products. During deep non-REM, the spaces between brain cells expand by about 60%. Cerebrospinal fluid flows through these expanded channels, washing away proteins like amyloid beta, one of the molecules implicated in Alzheimer's disease. This glymphatic clearance appears to operate similarly in men and women. The same expansion of intercellular space, the same flow of cerebrospinal fluid, the same removal of waste products. But because women tend to have more slow-wave activity, and because glymphatic clearance is most active during slow-wave stages, some researchers speculate that the female brain may have a slightly different pattern of nighttime waste clearance. More time in deep stages might mean more efficient removal of metabolic debris. This is still being studied. The data are preliminary, but the possibility is intriguing that the sex differences in rest architecture might serve a protective function, with the female brain's greater slow-wave activity providing a more effective maintenance system. Or it might not. Science is full of appealing hypotheses that don't survive further testing. What we know for certain is that the glymphatic system operates in both sexes, that it depends on the same stages of rest, and that its function declines with age in both men and women. The brain cleans itself every night, in both bodies, using the same channels, the same fluid, the same slow, patient rhythm of expansion and flow. Let's think about hormones and mood for a moment, because the biological rhythms of men and women don't just affect when they feel alert or drowsy. They affect how they feel emotionally. Serotonin, the neurotransmitter often associated with mood regulation, follows a circadian rhythm. Its levels fluctuate across the day in both sexes. But serotonin synthesis is influenced by estrogen, which enhances the expression of tryptophan hydroxylase, the rate-limiting enzyme in serotonin production. This means that in women, serotonin levels may fluctuate not only across the day but across the menstrual cycle. When estradiol is high, serotonin synthesis may increase. When estradiol falls, as it does in the late luteal phase and during menopause, serotonin production may decrease. This hormonal modulation of serotonin is one of the proposed mechanisms underlying premenstrual mood changes and the increased risk of depression during the menopausal transition. In men, serotonin follows a steadier rhythm. It still fluctuates across the day. It still responds to light, to diet, to exercise, but it isn't subject to the same monthly modulation. And yet, testosterone also influences serotonin. Low testosterone levels in men are associated with increased rates of depressive symptoms. The relationship is complex and bidirectional. Depression can lower testosterone, and low testosterone can contribute to depression. So both sexes experience hormonal modulation of their mood chemistry, but on different time scales. In women, the modulation cycles monthly. In men, it changes more slowly, across years and decades. Dopamine, the neurotransmitter involved in motivation and reward, also shows sex differences in its circadian pattern. Estrogen modulates dopamine receptor density in the striatum, potentially changing how the reward system responds across the menstrual cycle. The circadian rhythm of dopamine follows the same basic curve in both sexes, but the amplitude and sensitivity may differ depending on the hormonal environment. What this means is that the experience of motivation, of wanting, of finding things rewarding, may subtly shift across the month in women, not in a way that overwhelms conscious decision-making, but in the same quiet, background way that a 6-minute difference in clock speed reshapes the entire evening. The chemistry of feeling, tuned by the same rhythms that tune the clock, the temperature, and the tides of the night. There are aspects of male and female biology that converge in unexpected places. Consider the heart. The heart has its own intrinsic rhythm, generated by the sinoatrial node, a cluster of specialized cells in the right atrium. This rhythm is modulated by the autonomic nervous system and by circadian signals from the suprachiasmatic nucleus. Heart rate variability, the slight variation in time between consecutive heartbeats, follows a circadian pattern. It increases during the night when the parasympathetic nervous system is dominant and decreases during the day when sympathetic activity rises. This pattern is the same in men and women, but women tend to have slightly higher resting heart rates, about 5 to 8 beats per minute faster on average. And some studies suggest that women's heart rate variability shows greater responsiveness to the menstrual cycle, with shifts in autonomic balance during the luteal phase. The heart, like the brain, keeps time, and like the brain, it keeps time slightly differently in male and female bodies, while performing exactly the same essential function. Beat after beat, for decades, adjusting its rhythm to the hormonal tides that flow around it. Your heart is doing this right now, beating, adjusting, responding to signals from a clock you can't feel and hormones you can't see. Consider what happens to bone. Bone isn't the static structure most people imagine. It's a living tissue, constantly being broken down and rebuilt in a process called remodeling. Specialized cells called osteoclasts dissolve old bone. Other specialized cells called osteoblasts lay down new bone. This process follows a circadian rhythm, with bone resorption peaking at night. In women, estrogen plays a critical role in maintaining the balance between breakdown and rebuilding. When estrogen falls during menopause, bone resorption accelerates while bone formation stays the same or slows. The balance tips toward loss. This is why osteoporosis is more common in postmenopausal women. In men, testosterone supports bone density through a different pathway, partly through direct androgen effects on bone cells, and partly through the conversion of testosterone to estradiol by an enzyme called aromatase. As testosterone declines with age, bone loss also accelerates, though typically later and more gradually than in women. The same remodeling process, the same circadian rhythm of breakdown and rebuilding, but governed by different hormonal inputs that fail at different rates. Late in life, both men and women face the same challenge. A skeletal system losing density, its nightly maintenance cycles becoming less effective, its structural integrity slowly diminishing. Two different hormonal paths, one shared destination. Let me tell you about the kidneys for a moment, because even the kidneys have a circadian rhythm. The kidneys filter blood, produce urine, and regulate the balance of water and electrolytes in the body. And they do this on a 24-hour schedule. Urine production is highest during the day and lowest at night, which is why most people can get through the night without waking to use the bathroom. This rhythm is governed partly by vasopressin, a hormone released by the hypothalamus that tells the kidneys to retain water. Vasopressin levels rise at night, concentrating the urine and reducing its volume. In both men and women, this rhythm follows the same basic pattern. But with age, vasopressin signaling weakens, and the kidneys become less efficient at concentrating urine overnight. Nighttime urine production increases, and awakenings become more common. This happens in both sexes. It's one of the most consistent age-related changes in circadian function. But in women, the situation is complicated by changes in pelvic floor muscle tone after pregnancy and during menopause. The urge to wake may arrive not just because more urine is being produced, but because the structures that support urinary continence have changed. In men, prostate enlargement can create similar nighttime urgency through an entirely different anatomical mechanism. Different anatomy, different mechanisms, same result. the night becomes more interrupted as the decades pass. Let's return to the brain one more time, specifically to the question of how memories are processed during the night. Memory consolidation, the process by which short-term memories are converted into long-term ones, is one of the essential functions of rest. It occurs during both non-REM and REM stages, but through different mechanisms. During non-REM, the hippocampus replays the day's experiences in a compressed format, transferring information to the neocortex for long-term storage. This process is linked to the slow oscillations and spindles we discussed earlier. During REM, the brain appears to integrate newly stored memories with existing knowledge, finding connections and patterns that weren't apparent during waking hours. This is one of the reasons why insight and creative problem-solving often arrive after a night's rest. Both of these processes operate in men and women, the same hippocampal replay, the same neocortical transfer, the same REM stage integration. But because women generate more spindles, and because spindle density is linked to consolidation efficiency, there's a hypothesis, still being tested, that female brains may consolidate certain types of declarative memory slightly more efficiently during non-REM stages. And because progesterone reduces REM proportion during the luteal phase, there's a complementary hypothesis that the integration function of REM may be slightly diminished during that phase of the cycle. Two competing effects: more efficient consolidation in one stage, less time for integration in another. The net effect on memory performance across the menstrual cycle is still being debated. What isn't debated is that both sexes need the same basic architecture of the night to maintain cognitive function. Deprive a man or a woman of non-REM, and declarative memory suffers equally. Deprive either of REM, and emotional regulation deteriorates at the same rate. The same vulnerability, the same dependence, the same quiet necessity. Here's something about breathing during the night that connects to the differences and similarities we've been tracing. During non-REM, breathing becomes very regular. The respiratory rate slows. The tidal volume stabilizes. The diaphragm does most of the work, and the accessory muscles of breathing relax. During REM, breathing grows irregular. The rate fluctuates. The accessory muscles become paralyzed along with the rest of the voluntary musculature, and the brain's respiratory centers operate with less input from chemoreceptors, the sensors that monitor blood oxygen and carbon dioxide levels. This pattern is identical in men and women. But the susceptibility to breathing disruptions during the night differs by sex. Men are more likely to develop obstructive events, partial or complete collapse of the upper airway during rest. This is partly anatomical. Men tend to have narrower oropharyngeal airways relative to lung volume, and partly hormonal. Testosterone may promote fat deposition around the upper airway, while progesterone, which stimulates ventilatory drive, is largely absent in the male body. Women are relatively protected from these obstructive events during the reproductive years. Progesterone's stimulant effect on breathing, combined with different fat distribution patterns and a wider oropharyngeal airway relative to lung size, provides a degree of defense. After menopause, when progesterone falls, that protection diminishes. The rates of breathing disruption in women increase, approaching those of men. The converging theme again: different paths through the middle years, increasing similarity in the later ones. The air moves in and out of your lungs right now. Your diaphragm contracts and releases. Your respiratory centers in the brainstem adjust the rate and depth of each breath based on signals from the clock, from the blood, from the body. The same air, the same mechanism in every human chest. Let me tell you about what happens to the eyes during the night. Not to the retina's response to light, we covered that, but to the movements of the eyes themselves. During REM, the eyes move rapidly beneath closed lids. These movements aren't random. They're coordinated, conjugate movements, both eyes moving together, as if tracking objects in a visual scene. Some researchers believe they correspond to the visual content of dreams. The frequency, amplitude, and duration of these rapid eye movements appear to be similar in men and women. Whatever the brain is doing during REM, whatever scenes it's constructing, whatever memories it's replaying, it directs the eyes with the same basic program in both sexes. During non-REM, the eyes are mostly still. Slow, rolling eye movements occur during the transition from wakefulness to the first stage of non-REM, and then the eyes settle into relative quiet. This too is the same in both bodies, the same stillness, the same slow rolls at the boundary of wakefulness, the same rapid scanning in the depths of dreaming. Your eyes behind their closed lids will do this tonight. They'll slow, they'll still, and then when the brain enters its most active nocturnal state, they'll begin to move, tracing invisible paths across a scene that exists only inside your skull. The gut has its own clock. The enteric nervous system, sometimes called the second brain, contains about 500 million neurons, more than the spinal cord. These neurons generate their own rhythmic activity, governing the muscular contractions that move food through the digestive tract. This rhythmic activity follows a circadian pattern. Gastric motility is highest during the day and lowest at night. The production of digestive enzymes cycles with the clock. Even the permeability of the intestinal lining, how easily molecules pass through the gut wall into the bloodstream, fluctuates across 24 hours. In women, the gut clock is modulated by the menstrual cycle. Many women experience changes in digestion across the month. Bloating, altered motility, shifts in appetite. These are partly driven by progesterone's effects on smooth muscle tone and on the enteric nervous system. In men, the gut clock runs more steadily. Without the monthly hormonal cycling, the digestive rhythms hold closer to their daily baseline. Both systems are doing the same thing, though, timing digestion to match the hours of food availability and rest. The gut clock, like the master clock in the brain, is an ancient timekeeping mechanism that predates the evolution of mammals. It runs quietly, in both bodies, processing, absorbing, resting, on a schedule so old that its origins are lost in the deep history of animal life. There's a rhythm to the immune system that we touched on briefly, but deserves a closer look. Natural killer cells, a type of white blood cell that patrols the body looking for virus-infected cells and early tumor cells, follow a pronounced circadian rhythm. Their numbers in the bloodstream peak in the late evening and reach their lowest point in the early morning. This means the immune system's patrol is strongest at the time when the body is beginning its transition toward rest, and weakest in the morning, when cortisol is rising and suppressing inflammatory responses to prepare the body for the demands of the day. Women tend to have higher baseline counts of natural killer cells and a more vigorous innate immune response than men. This is one of the reasons women are generally more resistant to bacterial and viral infections. But it's also one of the reasons women are more susceptible to autoimmune diseases, conditions where the immune system attacks the body's own tissues. Estrogen enhances immune function. Testosterone dampens it. The same hormones that create the circadian and monthly rhythmic differences we've been discussing also create differences in how the body defends itself against pathogens. But the circadian rhythm of immune function, the daily rise and fall of natural killer cells, of cytokines, of inflammatory markers, follows the same curve in both sexes. The clock that tells the immune system when to activate and when to stand down is the same clock, ticking at the same pace, in every human body. The differences are in the amplitude, the height of the peaks, the depth of the valleys, not in the rhythm itself. Let me share something about how the two hemispheres of the brain behave during rest. There's evidence that in the early stages, particularly in unfamiliar environments, one hemisphere of the brain remains slightly more vigilant than the other. This phenomenon, studied most extensively in birds and marine mammals, appears to exist in a mild form in humans as well. When a person rests in a new place for the first time, the left hemisphere shows more responsiveness to external stimuli than the right. The brain keeps one side slightly on guard, ready to wake if something unexpected occurs. This effect diminishes with familiarity. By the second night in a new environment, the asymmetry is reduced. And it appears to be similar in men and women. The same protective mechanism, the same hemispheric vigilance, the same adaptation over time. It's one of the most ancient survival strategies embedded in the resting brain. A remnant of the time when rest was dangerous, when predators moved in the dark, and the body needed to maintain some connection to the outside world even as consciousness withdrew. That ancient circuit is still active in you. Tonight, wherever you are, your brain will decide how safe the environment is and calibrate its hemispheric vigilance accordingly. If you're in a familiar place, both hemispheres will descend together into their shared rhythm. If you're somewhere new, one side will hold back, just slightly, keeping watch while the other rests. The same in men, the same in women, a shared inheritance from a time before there were men and women at all. Here's a fact about growth hormone that connects to the rhythms of the night. Growth hormone is released in pulses during the first half of the night, predominantly during deep non-REM stages. These pulses are critical for tissue repair, muscle recovery, and metabolic regulation. In both men and women, the largest pulse of growth hormone typically occurs within the first 90 minutes of rest. This is when slow-wave activity is at its peak for the night. The total amount of growth hormone released during the night declines with age in both sexes. In women, this decline is compounded by menopause, which reduces the deep slow-wave stages during which growth hormone is preferentially released. In men, the decline is more gradual, tracking the slow reduction in slow-wave activity that accompanies aging. But in young adults, the pattern is strikingly similar. The same first cycle pulse, the same association with slow-wave stages, the same critical role in recovery and repair. Growth hormone doesn't care about sex. It cares about the depth of the oscillation. It cares about the amplitude of the slow waves. It cares about the quiet signal from the hypothalamus that says, now, release, repair. That signal will come tonight, in your body, During the deepest part of the first cycle, a pulse of growth hormone released into the blood, carrying instructions for repair to every tissue it touches. The same pulse in every resting body, since before humans knew what hormones were. Let me tell you about dreams, not the content that varies endlessly, but the biology of dreaming. During REM, the brain becomes almost as active as it is during waking. The prefrontal cortex, the region involved in logical reasoning and decision-making, becomes relatively quiet. The limbic system, the emotional centers, lights up with activity. This is why dreams are often emotionally vivid but logically incoherent. The emotional brain is running. The logical brain is dimmed. Both men and women show this same pattern. The same prefrontal deactivation, the same limbic activation, the same increase in acetylcholine, the neurotransmitter that drives REM. What differs is the hormonal context in which these dreams occur. During the luteal phase, when progesterone has reduced the proportion of REM, women may experience fewer dream periods per night. During the follicular phase, when hormones are at their lowest, REM rebound may occur, the brain compensating for lost dream time with more intense or more frequent REM episodes. Men's dreams occur in a more hormonally steady context. The nightly fluctuations in testosterone don't substantially alter the frequency or intensity of REM episodes from one night to the next. The theater of the dream operates by the same rules in both bodies. The same actors, the limbic system, the visual cortex, the motor cortex sending movement commands that are blocked by the brainstem before they reach the muscles. The same paralysis that keeps the dreaming body still, while the dreaming mind moves through impossible spaces. But the schedule of performances differs. In one body, the schedule shifts with the month. In the other, it holds steady. Same theater, different playbills, same darkness in the auditorium. There's something worth knowing about pain perception and how it relates to the body's rhythms. Pain sensitivity follows a circadian pattern. It tends to be lowest in the afternoon, when cortisol is declining but still present, and when body temperature is near its daily peak. It's highest in the early morning hours, when cortisol is just beginning its rise and the body's anti-inflammatory defenses are at their weakest. This rhythm is similar in men and women, but women, on average, show greater sensitivity to several types of experimentally induced pain, including thermal and pressure. And this sensitivity fluctuates across the menstrual cycle, typically increasing during the late luteal phase when progesterone and estradiol are falling. The mechanism involves both peripheral and central factors. Estrogen modulates the expression of opioid receptors in the brain and spinal cord. When estrogen is high, the endogenous pain relief system may function more effectively. When it falls, pain sensitivity increases. This creates a monthly rhythm of pain sensitivity layered on top of the daily circadian rhythm. Two oscillations interacting: the 24-hour cycle of cortisol and temperature, the 28-day cycle of estrogen and progesterone. Together, they create a complex pattern of vulnerability and protection that shifts from hour to hour and week to week. Men experience only the daily rhythm. Their pain sensitivity still fluctuates, still peaks in the early morning, still reaches its lowest point in the afternoon. But without the monthly modulation, the pattern is more predictable. Two versions of the same rhythm, one simple, one complex, both real, both the body's way of keeping time with its own vulnerability. The skin has a circadian rhythm too. Cell division in the skin's basal layer, the layer that produces new skin cells to replace the ones constantly shed from the surface, peaks during the night. The skin renews itself most actively while the body rests. This makes sense from an evolutionary perspective. Cell division is a vulnerable process. The DNA must be unwound, copied, and reassembled. Errors during this process can lead to mutations. Doing this work during the night, when UV radiation from the sun is absent, minimizes the risk of radiation-induced DNA damage during replication. This rhythm is the same in men and women. The same nighttime peak in cell division, the same protective timing, the same daily renewal of the body's largest organ. But the skin itself responds differently to hormonal changes across the menstrual cycle in women. Sebum production, the oily secretion that moisturizes and protects the skin, fluctuates with hormonal levels. This is why some women experience changes in skin texture and breakouts at predictable points in their cycle. In men, sebum production is more stable across time, driven by the steadier testosterone rhythm. The skin breathes, it renews, it protects, and it does all of this on a circadian schedule that's shared between every human body, while responding to the unique hormonal weather of each individual. Let me tell you about the connection between these biological rhythms and longevity. There's growing evidence that the strength of circadian rhythms, their amplitude, their stability, their resistance to disruption, is associated with health and lifespan. Animals with strong, high-amplitude circadian rhythms tend to live longer than those with weak or disrupted rhythms. In humans, age-related weakening of circadian rhythms is associated with cognitive decline, metabolic dysfunction, and increased mortality risk. And this association holds for both men and women. But the paths to circadian disruption differ by sex. In women, the abrupt hormonal changes of menopause can acutely destabilize circadian function. Hot flashes, nighttime awakenings, and shifts in melatonin timing all contribute to a period of circadian fragility that can last several years. In men, the disruption is more gradual, a slow erosion of circadian amplitude over decades as testosterone declines. Both paths lead to the same outcome, a circadian system that's less precise, less capable of maintaining the sharp distinctions between day and night that characterize youth. And yet, some individuals maintain strong circadian rhythms well into old age. They're the chronobiological equivalent of centenarians, people whose internal clocks remain precise and powerful despite the passage of time. What protects them isn't fully understood. Genetics plays a role. So does regular light exposure, consistent meal timing, physical activity, and social engagement, all of which provide time cues that help maintain the circadian system's alignment with the external world. These protective factors work in both sexes. The same strategies, the same benefits, the same quiet reinforcement of the body's oldest timekeeping system. Let's talk about something that might not seem related at first, but is deeply connected to everything we've discussed: the biology of attachment. When two people form a close bond, romantic, parental, or between close friends, their physiology begins to synchronize. Heart rates align, breathing patterns coordinate, and circadian rhythms over time converge. Partners who live together tend to develop increasingly similar circadian timing. Their melatonin onset shifts toward a shared middle point. Their cortisol rhythms begin to mirror each other's. Even their temperature curves, those quiet daily oscillations that govern so much of the body's function, begin to align. This synchronization occurs between partners of any sex combination. It isn't dependent on the specific hormonal environment of either individual. It's a property of proximity, of shared light exposure, of coordinated meal times and activity patterns. The circadian system is social. It responds not only to light and hormones, but to the presence of other bodies. And this social synchronization may have deep evolutionary roots. In species that live in groups, coordinating activity patterns improves collective safety and efficiency. The circadian clocks of group members drift toward alignment, ensuring that the group wakes together, forages together, and rests together. We carry this same circuitry, the same responsiveness to social timing cues, the same tendency to synchronize with those around us. Tonight, if someone you love is nearby, your clocks are quietly adjusting to each other, not consciously, not deliberately, but through the simple fact of shared light, shared meals, shared time. Two clocks, different in their individual calibration, but pulling gently toward the same rhythm. And now we're approaching the end of our journey through the biological rhythms of men and women. The differences we've looked at tonight in clock speed, in light sensitivity, in hormonal modulation, in pain perception, in immune function, in developmental timing, are real and measurable. They have practical consequences for health, for well-being, for the experience of daily life. But they're all variations on a single theme. The human body keeps time. It keeps time with extraordinary precision and complexity. It uses multiple overlapping systems, circadian, infradian, ultradian, circannual, to synchronize its internal processes with the external world. Men and women share every one of these timekeeping systems. Every one. The suprachiasmatic nucleus, the pineal gland, the peripheral clocks, the hormonal cascades, the neural pathways that carry light from the retina to the clock. The differences lie in the modulation, in the hormonal signals that tune each system to a slightly different frequency, in the developmental rhythms that set those hormonal patterns during adolescence, in the monthly cycles that reshape the female night while the male night holds steady. And in the end, at the far end of the lifespan, even these differences fade. The clocks converge, the hormonal modulations weaken, and what remains is the shared biology of aging, the quiet slowing of rhythms that once ran strong. The body's rhythms aren't separate in men and women. They're the same rhythms played in different keys. And as the decades pass, the keys slowly merge, until the melody is recognizable as one. You've been listening to a story about the time your body keeps without your knowledge, the oscillations you can't feel, the cycles you've never seen on any clock. Right now, melatonin is rising in your blood. Your core temperature is falling. The suprachiasmatic nucleus, that grain of rice-sized cluster behind your nose, is sending its nighttime signals outward through the brain. Your peripheral clocks are synchronizing. Your kidneys are concentrating your urine. Your skin cells are beginning their nightly division. Your immune system is adjusting its patrol. All of this is happening in your body right now, whether you're a man or a woman, whether your clock runs at 24.09 hours or 24.19. The rhythm continues. The heart beats, the breath moves, The blood carries its signals from organ to organ, from clock to clock, from one quiet cycle to the next. And somewhere within this vast and patient counting, within these nested rhythms that began before you were born and will continue as long as you live, the body does what it has always done. It keeps time. It keeps time with the turning of the earth, with the pull of hormones and the glow of light and the presence of those nearby. And tonight, as it has every night of your life, it will carry you gently from wakefulness into something quieter, something older than any clock, something shared by every human body that has ever drawn breath on this small, spinning world. The rhythms continue. The light fades. And rest, when it arrives the same way it always has. Quietly, patiently, in its own time.